Have you ever stopped to think about the invisible world living in your food? I'm talking about microbes. These tiny organisms are the secret chefs behind some of the most incredible flavors we know and love. Today, we're going to see how cracking their genetic code is literally unlocking the future of what we eat.
So think about it. That amazing tang in sourdough bread, the satisfying crunch of kimchi, or even just a simple yogurt. What is it that gives them that special something? Well, it's not just the ingredients you can see. It's an entire bustling city of microscopic life, all thanks to fermentation driven by bacteria. But what happens when you zoom in, like really zoom in on those bacteria?
Well, our story is going to take us all the way to southeast Nigeria, to a food seasoning called Ogiri-Okpei. This stuff is made by fermenting the seeds of the African mesquite tree. And let me tell you, it creates this incredibly powerful, pungent aroma that is the heart and soul of so many traditional dishes. And the secret, of course, is all in the microbes.
Scientists who are looking into this very seasoning found two key players, two bacterial strains that are about to become the heroes of our story. They're almost identical, but as you're about to see, in the world of microbiology, almost is a very, very big word. Okay, so let's meet them. On one side, we've got the Onitsha strain, also known as KCA4, and on the other, the Nsukka strain, KCA5. Now get this, they are the exact same species, Lactiplantibacillus plantarum, and they both came from the same food, Ogiri-Okpei. The only real difference? They were found in two different cities, Onitsha and Nsukka, about 144 kilometers apart. You can kind of think of them as two siblings who grew up in different towns.
And right there, that's our central mystery. They're practically twins, but do they act the same? What tiny differences could possibly exist between them? And more importantly, why on earth should we care? Well, the answer could have huge implications for everything from our health to the future of our food.
To solve this mystery, you gotta look at the source code. We're talking DNA. So let's crack open their genetic cookbook and see if the recipes are actually different.
You know, a microbe's genome is basically its operating system. It's the complete set of instructions for building the microbe and for every single thing it can possibly do, from what it likes to eat to how it defends itself. By comparing their genomes, we can start to see what really makes each of these strains tick. All right, first up to the plate is the Onitsha strain, KCA4. After sequencing its DNA, scientists found its genome is just a little over 3.3 million base pairs long. That's 3.3 million letters in its genetic instruction manual. And what about its sibling from Nsukka, KCA5? Well, its genome is just a tad smaller, coming in at just over 3.2 million base pairs. So right off the bat, that's our first clue. The Onitsha strain's cookbook is a little thicker. It's got more genetic code. So, what does that extra genetic information actually do? Well, a big part of it is about their metabolic toolkits. This chart here shows the number of genes for something called enzymes. Just think of them as specialized kitchen gadgets for breaking down carbs and sugars. The Onitsha strain, KC4, has 101 of these tools. Its sibling from Nsukka only has 95. It's a small difference, but it's a significant one. But this is where it gets really fascinating. Just like us, bacteria can get sick. They get infected by viruses. And sometimes, the DNA from those viruses gets stitched right into the bacterium's own genome. These little genetic hitchhikers are called prophages, and they can actually give the bacteria totally new abilities. It's like carrying around viral baggage that might just contain a secret superpower. And here's where we find another huge difference. The Onitsha strain, KCA4, has two intact sets of this leftover viral DNA. But the Nsukka strain, KCA5, it has three. That extra piece of viral baggage in the Nsukka strain could completely change how it behaves. So it's pretty clear these two siblings have led very different lives, at least genetically. Okay, so we've looked at the blueprints, we've seen the genetic differences on paper, but what does any of this mean in the real world? It's time to put these microbes to the test and see what they can actually do. So the first test is all about stickiness. For a microbe to be a good probiotic, one that's actually beneficial for your health, it needs to be able to stick to the cells in your gut. So the researchers set up this really cool test. They grew a layer of human intestinal cells, introduced each bacterial strain, and then after a while, they just washed them off to see how many managed to hang on. And the results? They were crystal clear. The Nsukka strain, KCA5, was the undisputed champion of stickiness. Almost 7% of its cells were able to adhere to the intestinal lining. The Onitsha strain, KCA4, wasn't quite as grippy, with a rate of about 4.4%. And let me tell you, this is a huge deal. That extra stickiness makes KCA5 a much more promising candidate for becoming a future probiotic.
But, being a good probiotic isn't just about sticking around. It's also about protecting its home turf. So for the next test, the researchers put our two strains up against some common bad guys, some harmful pathogens, to see who was the better fighter.
And the results here were pretty interesting. Both strains were just as good at fighting off E. coli and Salmonella. But in the fight against Staphylococcus aureus, which is a really common and sometimes dangerous bug, the on-a-shell strain, KCA4, actually showed slightly higher activity. So, while KCA5 is stickier, KCA4 might be a slightly better bodyguard against that one specific threat.
So, what do we have? We've got a stickier microbe and a slightly better fighter. Now, these might seem like really tiny differences, but they open up a whole universe of possibilities. Let's look at why this deep dive into a traditional food seasoning actually matters for all of us.
I mean, the potential here is just astounding. KC5's incredible stickiness makes it a top-tier candidate for new, more effective probiotics. The antimicrobial powers of both strains could be used as natural food preservatives, replacing artificial chemicals. And they're unique genetic toolkits for creating flavor. They could be used to engineer entirely new tastes in the food industry. We're even talking about creating functional foods that don't just feed you, but might actively improve your health by producing compounds like GABA, which is linked to reducing anxiety. And hey, don't just take my word for it. The scientists who did all this work agree. They concluded that the unique profiles of both of these strains have, and I quote, potential industrial fermentation utility. That's science talk for, this is going to be a very big deal. So this whole tale of two microbes, born from the same food but shaped by totally different lives, it just reveals a universe of potential that's been hiding in the most traditional places all along. It really makes you wonder, doesn't it? What other microscopic secrets are just fermenting away in our food, waiting for us to find them?